Hi, I'm Erin, and my research is about unraveling the patterns of mutational signatures in pediatric brain tumors. I'm a rising senior at Lowndes High School in Georgia, and I hold leadership positions in several clubs in STEM, and I am a USA Biology Olympiad semifinalist, Chemistry Olympiad National Qualifier, and I presented my research at several conferences. I aspire to be a pediatric neurosurgeon. Mutational signatures are an emerging global health topic that has garnered significant attention in recent years. So, this presentation aims to elucidate why mutational signature research has become a critical global health issue. Mutational signatures are distinct patterns of genetic mutations, and they provide clues about the underlying biological processes. How? Let's consider cancer. Do you know how cancer develops? Cancer is a significant global health concern and is caused by mutations in genes. Each mutational process imprints a characteristic pattern of mutations on the tumor genome, which is known as a mutational signature. Let's take a look at Figure A, which shows mutational signatures associated with tobacco smoking in human cancer. The pattern of mutational signatures is observed in tobacco smoker cancers in Figure B. For instance, in lung cancer, the tobacco-associated signature is typically characterized by a high frequency of C to A mutations. Therefore, mutational signatures enhance cancer prognosis prediction and potentially support treatment by revealing cancer risk factors. This flowchart summarizes how mutational signature research proceeds. That is, when cancer is developed due to the mutational process, it imprints a distinct pattern of mutation called mutational signatures. The researchers decipher such patterns of mutations and identify factors. This knowledge is crucial for developing effective prevention strategies and targeted therapies. Therefore, mutational signature research is critical, impacting disease etiology, personalized medicine, public health, and cancer research. Early cancer detection via mutational signature analysis enhances survival rates. However, limited research exists on the impact of clinical features like age and sex on mutational signatures in pediatric brain tumors, hindering cancer prevention efforts. So, my research aims to investigate the impact of age and sex on mutational signatures in pediatric tumors to understand cancer development across childhood age groups. I developed 3 research questions for my project regarding the association between the clinical features of age and sex with mutational signatures. I developed the corresponding null hypothesis and alternative hypotheses. My research was conducted through the following steps. First, I collected whole genome sequencing-based TSV files from the ICGC data portal, and the data consists of mutational signatures, specimen histology, and clinical information. Next, I grouped data by tumor type and by clinical features like age and sex. I analyzed mutational signatures in 2 comparing groups: the childhood group, ages 3 to 17, and young adults, ages 18 to 49. And I determined the relationship between age and sex regarding mutational signatures. Finally, I investigated the relationships between sex-specific mutational signatures and non-sex-specific mutational patterns. I collected data using R and then analyzed data using a Wilcoxon rank sum test, and I used box plots to visualize my data. The findings of my research demonstrated that the childhood group shows a higher frequency of somatic mutations associated with the analyzed signatures, as depicted in figures A, B, C, and D. Notably, signatures SPS18 and ID9 in figures B and D show statistically significant differences, with the p-value being less than 0.05 between the groups, providing strong evidence against the null hypothesis. Regarding the sex factor, my research found that the SPS40 mutational signature in pilocytoacytoma was strongly associated with female patients, with a significantly higher number of somatic mutations observed in females compared to males. I determined the relationship between age and sex clinical features regarding mutational signatures, and the figure illustrates distinct somatic mutational profiles of mutational signatures in female pilocytic astrocytoma patients, for instance, signatures SPS40, shown in light green bar. I investigated the relationship between sex-specific mutational signatures and non-sex-specific mutational patterns, and the correlation plot shows this relationship between mutational signatures, where the X and Y axis list the analyzed mutational signatures, forming this symmetric grid, where the red indicates a strong positive correlation. And the figure shows a strong correlation between signatures SPS40 and others like ID5 and SPS1, as evinced by the more reddish color. So this study suggests the importance of conducting further research to explore the relationship between SPS40 that's associated with the sex factor and other factors that are related to ID9 and SPS1. Investigating the potential connections and interactions between these mutational signatures can provide valuable insights into how sex-related factors may influence mutational processes in conjunction with other molecular factors. Overall, my project tackled the challenge of the relatively new association between age and sex clinical features on mutational signatures in pediatric brain tumors, and my results underscore the role of these clinical features in mutational signatures and the need for further consideration of these features in future pediatric cancer research. My findings are a significant step towards tumorigenesis and cancer evolution in pediatric brain tumors. My feature research will investigate how mutational signatures would be associated with germline variants, and it'll be through the following stages. To first investigate the identified mutational signatures and examine factors like sex-specific genetic factors, and examining the characteristics of signatures whose etiologies are unknown. It's my hope to provide valuable insights into cancer evolution in pediatric brain tumors. I'm deeply grateful for my mentor, Dr. Faroki. Thank you.